Okay. Hi everybody. My name is Robert Brocklehurst. I'm from the University of Manchester, and today I'm going to be talking about vertebrae, ribs, and lungs in archosaurs. So, kick things off, I'm just going to take a moment to talk about how great birds are. They are the most diverse group of living tetrapods. They have 10,000 species. You find them all over the world. They're incredibly disparate. A disparity I've attempted to condense into 4 pictures. And so a lot of people have focused a lot of attention on exactly why they're so successful and exactly why they're so diverse. Particularly when you consider that they're part of this broader group, the Archosauria, which literally means ruling reptiles. And their living sister group, the crocodiles, are not very diverse. They have about like 20 species, maybe it's probably a bit higher than that, but it's not, they're 10,000, it's quite a big difference. And also where this diversity of birds came from in terms of the fossil record and in terms of evolution, because everyone's favourite group of stem birds, the dinosaurs, eventually gave rise to modern birds. So I'm interested in where the adaptations that have led to the success of birds came from in evolutionary time. And one of the things that people suggest as a reason for the success of modern birds is their respiratory system, which is very derived, is very efficient, is very good at getting oxygen out of the air. So you compare the respiratory system, the lungs of a bird, to those of a crocodile. The crocodile has a lung like you expect a lung to look like. It's this organ that inflates when you breathe in and deflates when you breathe out and carries out gas exchange and fills in the whole thoracic cavity. Birds' lungs don't look like that. Birds' lungs only do gas exchange. All a bird's lung does is get oxygen out of the air. A bird's lungs do not increase or decrease in volume when they breathe. The process of getting air through the lung is handled entirely by this massive air sac system that is a particular avian innovation. And this air sac system has been the focus of a lot of attention in the fossil record because it's associated with postcranial pneumaticity, i.e. air-filled bones, not in the skull. And you see pneumatic features, like in this duck, you see these holes in the bone, and you also see this in dinosaur fossils. And an air sac system has been posited on this basis in pterosaurs, sauropods, theropods, all sorts of archosaur groups. But the presence of air sacs doesn't necessarily tell you anything about the structure of the lung itself. And so that's what I'm interested in. Is there a direct way you can tell what the lung itself was like? And the answer turns out to be very helpfully, well, hopefully at least, yes. So for these figures, imagine you have a crocodile or an ostrich on this table, belly up, and you've just cut it open, and you've pulled the lungs out, which is what these two are here. So these are crocodile lungs, and you can see that they're nice and smooth on top. And this part here, so if you're looking, imagine you're looking down into the now empty body cavity of this crocodile from which you have just pulled its lungs, then you are looking at the sort of bottom, like the inside of the ribcage here, and you'll notice that what people call the thoracic ceiling is nice and smooth. If you do the same thing to your ostrich, you cut it open, you pull its lungs out, you can see that it's got these big dents in the lung tissue. And that's from like the ribs actually embedding themselves into the lung, which people think is an adaptation to rigidity and helps maintain lung structural integrity. And if you look at the thoracic ceiling of an ostrich or any bird, then it's got these big ridges and these furrows from where the ribs attach to the vertebral column. And so that joint, where the ribs attach to the vertebral column, is actually really important and is a very useful indicator of lung structure in archosaurs. So these two joints here, the parapophysis and diapophysis, which for the rest of this talk I will simply refer to as the pink one and the blue one. In birds, the pink one stays underneath the blue one for the entire vertebral series, whereas in crocodiles, the pink one starts off underneath the blue one, but then sort of migrates upwards and effectively gets out of the way of the lungs so that they can expand and contract freely underneath the ribs and the vertebrae. So my question, though, is that this has been studied quite a lot before in living and fossil archosaurs, but it's all been very descriptive and no one's really put numbers on any of this. So my question is, how reliable is it, especially when you actually try and quantify this joint structure and how it relates to lung morphology? And so then, extending that back into the fossil record, what was the lung structure of dinosaurs like, and other archosaurs eventually, but focusing on dinosaurs for today. So, to do that, I went to a load of museums and collected loads of data on living and fossil archosaurs using a variety of methods, CT scanning, photogrammetry, structured light scanning. Unfortunately, I didn't quite get as much dinosaur data as I wanted, so what I ended up doing was screenshotting all of my 3D models and then searching through the literature for illustrations of dinosaur vertebrae. So just this is like a little snapshot of my sample. So on the top you have the dorsal series of an ostrich. In the middle you have the dorsal series of an American crocodile. And then at the bottom you have Diplodocus. Those are actually Dippy's vertebrae, if anyone's interested. I don't just have these three. I have lots of other birds and crocs and dinosaurs, but that's just a little snapshot of the sample that I've got. And then I subjected them to geometric morphometrics analysis, which has been explained before. So I used this set of landmarks, which I borrowed from another paper, which was very well received. So if anyone has any questions about the numbers and positions, etc., this is an established scheme that I used. And like I said, it was just two-dimensional from the front, so then you can trace not only the motion of what would be the pink joint upwards, but also the motion of the pink joint outwards. So, on to my results. This is a little refresher of what the lungs and the joints look like in a bird and a crocodilian. And now onto morphospace number 1. Well, the first version of the morphospace anyway. So this is my vertebral morphospace. You have the birds in red and the crocs in yellow and various dinosaur groups in the other colours. And so what you can first thing you can kind of see is that dinosaurs are just off doing their own thing, really. They're not actually plotting out particularly closely to either of the extant Archosaur groups. Which was, actually, as I was collecting the data, I kind of figured this would happen. It's not entirely helpful, though. And I'm just going to quickly talk about what's going on with the crocodiles, especially, because they overlap with birds a little bit. But that's all part of the variation you get along the vertebral column in crocodilians. So they start, because as I said, they start off with the pink one underneath the blue one. And that's what you're seeing up here. This is the anterior part of the crocodilian vertebral column, and then as you get further back, they all sort of drift off down here. What does that actually look like when you look at it in terms of shape change and in terms of joint positions? So these are the mean shapes of each group. So that's the mean crocodilian vertebra, the average bird, the average dinosauromorph, average theropod, sauropod, and ornithischian. And so the first principal component axis, so left and right, seems to mostly separate dinosaurs from the extant things. And that's mostly due to the angle of the transverse processes. Steeply angled versus flat. Whereas PC2, which separates out dinosaurs and birds from crocodiles, is about the position of the pink joint relative to the blue one. So up here, it's below it. Up here, it's sort of in line with it. It moves upwards as you go down this graph here. So I was a bit put out by this result, to be honest, because I thought, I can't actually say much about dinosaur lung structure based on these results because they don't nicely plot with either birds or crocodiles. So what I did was I looked at Euclidean distance metrics, basically trying to figure out, which one are they more like? Which one are they closer to? And when you do that, they're all consistently closer to birds than to crocodiles. So this is, that's just the same graph as we had on the previous slide. And then this is a chart of just Euclidean distance from the center of the group to either birds or crocodiles. And the distances are always lower to birds than to crocodilians. So despite the fact that they're not in the same part of space as birds, they're still closer to birds than to crocs. And, well, what does all that mean? What does any of this mean? So, this hopefully illustrates something about the evolution of the dinosaurian respiratory system beyond the data that we already have about pneumatization and about air sacs. And I'm going to look at integrating these two halves more further on in my PhD. Once I have more dinosaur data, I'm also going to be looking at expanding this and doing a full 3D analysis, as was the original intention, and putting more of this in a phylogenetic context. Because ideally, I want to understand the origin of like the high performance lifestyle that is modern birds. They have very high metabolic rates. They are capable of very prolonged periods of very highly demanding aerobic activity, i.e. flying. And this physiology came from somewhere. And I'm interested in the assembly of the traits that have led to them being like a respiratory system that is capable of sustaining that metabolism and sustaining that mode of locomotion, and effectively what made birds birds. So with that, just to sum up, vertebral morphometrics is capable of separating out the living taxa. They are different. Dinosaurs don't actually map particularly closely to one or the other. They're sort of off doing their own thing. But based on the Euclidean measures and also still the fact that they plot out not with crocodiles, that leads me to suggest that they have a sort of heterogeneously partitioned lung. So the lungs might have been, parts of the lung might have been more adapted to gas exchange and parts of them might have been more ventilatory in their function, but they didn't have this distinction that you have in birds between air sacs and lungs. They had this sort of intermediate system. And with that, I'd just like to thank a few people. The triumvirate of my supervisors, Bill, John, and Phil, and that's not the right button. There we go. Bill, John, and Phil, Emma Shatner, my biggest external collaborator, various people from all of the museums who let me come and look at their stuff, and all of you for listening. Does anyone have any questions?